Something like 400,000 people are killed every year by malaria, and more than 90% of these people, up to 95% by some estimates, are killed by just one particular strain. Malaria is caused by the Plasmodium malariae amoeba, which gets inside of and destroys red blood cells. There are many different Plasmodium malariae subspecies, some being more or less pathogenic than others. Most of these subspecies can cause a mild form of malaria, but some are much more aggressive. And the most dangerous, without a doubt, hands down, the most dangerous, is the Plasmodium falciparum, which is the one strain responsible for causing 90 to 95% of annual global malaria deaths. A new study sought to examine exactly why this particular subspecies is so dangerous, and the research began with a series of enzymes called kinases. Now, the two important things to understand about kinases are that they can, 1, transport molecules around inside the cell, and 2, they can facilitate the phosphorylation of other biomolecules, activating enzymes and altering the epigenetics of DNA, and this can affect the regulation of the expression of the affected genes. Once the Plasmodium falciparum amoeba has infected a host blood cell, it appears to export 18 different kinases, which causes a downstream cascade of effects within the host blood cell. One of these changes to the host cell involves making it much more rigid and inflexible, which will impair its movement through the vasculature. This can lead to blood clots that damage organs and other tissues. If a blood clot blocks a vein, then all of the tissue downstream that was, nourished by this vein, it's not getting nutrients or oxygen anymore. And a lack of oxygen is the quickest way to kill off a large number of cells. Another change involves the increase in adhesin proteins on the blood cell surface. So not only is the red blood cell stiffer and less flexible, it's also stickier and will bind to cells on the inner surface of blood vessels, potentially leading to blockages. At the very least, the infected blood cells will fail to circulate throughout the body, and they won't go through the kidneys or the spleen, and it's harder for the immune system to find it. This is a really important aspect of malarial pathology, because if the pathogen can sneak inside of a host cell and alter it so as to hide from immune cells, it'll be that much harder to get rid of it, to get rid of the infection. It'll be that much harder for the immune system to destroy it and remove all traces of malaria from the body. This is the primary reason why infection with Plasmodium falciparum can be so deadly. Hugo Belda is a PhD student in the Signaling and Apicomplexan Parasites Laboratory at the Francis Crick Institute, and he was a co-lead author of the paper. Mr. Belda said, quote, "These kinases are not released by other strains of malaria that infect humans, so we predicted that they're some of the factors that makes this species deadlier," unquote. Helid Davies was another co-lead author of the research, and she said, quote, "This malaria parasite species is able to use a number of different variants of the same protein to make red blood cells sticky. So if the body develops antibodies that stop one variant from working, the parasite can simply switch to another one, leading to a constant arms race," unquote. In their study, they identified which kinases are involved in these particular changes in the host cell. For example, the FIKK4.1 kinase is involved in both the, quote, "rigidification of the erythrocyte cytoskeleton and trafficking of the adhesin and key virulence factor PFEMP1 to the host cell surface," unquote. So what they're saying here is that, that particular kinase, which has been labeled FIKK4.1, that particular kinase affects both the stiffening of the cell and the movement of these adhesin proteins and other infectious factors to the surface of the host cell to help it stick to the side of a blood vessel to hide from the immune system, where it will then quietly secrete chemicals into the bloodstream. The researchers' ultimate conclusion, summed up in a single sentence, is that, quote, "This establishes the FIKK family as important drivers of parasite evolution and malaria pathology," unquote. So this research is really important because they've basically found a critical group of enzymes responsible for the stunning lethality of Plasmodium falciparum. These are now targets for drug researchers who will try to develop something that can interfere with the expression or the activity of this entire FIKK family of kinases. We can make drugs that might block these genes from being expressed. We can potentially make drugs that inhibit the activity of the entire family of enzymes by targeting some family-specific trait or feature of the polypeptide. Although this might be tricky because there are all kinds of other healthy kinases that our bodies need, and a drug that targets some chemical component of a kinase might accidentally have overlap with a healthy kinase, and inhibiting that would hurt the patient. And it's possible that there might be some other approach that involves the FIKK family somehow. Now, this is how medical science happens. This is how new medicines are made, how things are discovered and developed. And this is how diseases that once killed millions can potentially be controlled and subdued.